Did you know? Right now, as you're watching this video, enzymes in your body are driving millions of chemical reactions every second to keep you alive. Stay tuned for more about enzymes and how they work inside your body. Thank you. Let's go.
Right now, millions of life-saving chemical reactions are happening inside your body. Without enzymes, life would slow to a stop. Enzymology is the science of these biological tools, the tiny proteins that keep every cell alive and functioning. Enzymes are biological catalysts. They speed up reactions without being used up or destroyed. Almost all enzymes are proteins, long chains of amino acids folded into precise three-dimensional shapes. Shape is everything. If an enzyme loses its shape, it loses its power. Enzymes are extremely specific. Each enzyme works on only one particular molecule, its substrate. Their efficiency is extraordinary. Enzymes can speed up reactions by millions of times. And after each reaction, the enzyme is released, unchanged and ready to work again. Every chemical reaction needs an energy push, called activation energy. Enzymes lower this energy barrier, making reactions easier and faster. First, the substrate enters a special pocket on the enzyme, the active site. They form a temporary enzyme-substrate complex. The enzyme bends and stresses the molecule, breaking or forming chemical bonds. The products are released, and the enzyme returns to its original shape. This flexible interaction is called the induced fit model, like a glove molding around a hand. Most enzyme names end with "ase", often based on what they act on or what they do. Scientists group enzymes into 6 major classes based on the reactions they catalyze. These include oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Oxidoreductases transfer electrons. Transferases move chemical groups. Hydrolases break bonds using water. Lyases break bonds without water. Isomerases rearrange molecules. Ligases join molecules together using energy. Enzymes only work under specific conditions. Change the environment, and their activity changes. As temperature increases, so does the speed of the enzyme reaction, up to an optimum point. Beyond this point, high temperatures cause the enzyme activity to decline. Too much heat denatures the enzyme, permanently destroying its shape.
pH also matters. Each enzyme works best at a specific acidity level.
Amylase prefers neutral. Pepsin works in strong acid. Arginase works in alkaline conditions. If pH changes too much, electrical charges in the active site shift, and the enzyme stops working. Increasing substrate speeds reactions until every enzyme is busy. This is called saturation. Sometimes enzymes must be slowed or stopped. This control is called inhibition. Competitive inhibitors mimic the substrate and block the active site. Adding more substrate can overcome competitive inhibition. Non-competitive inhibitors bind elsewhere, changing the enzyme's shape. This allosteric change prevents the substrate from fitting, no matter how much substrate is present. Irreversible inhibitors bind permanently disabling the enzyme. Deadly poisons like cyanide work this way, shutting down cellular energy instantly.
Life depends on enzymes. Their shape controls their power, and without them, life cannot exist.
Please comment on this video, like, share and subscribe to help us grow this channel. Thank you so much.